Welcome to Chapter 20, Part 2. In this half, we're going to go over tests used to guide chemotherapy, resistance to antimicrobial drugs, antibiotic safety, effects of combinations of drugs, and the future of chemotherapeutic agents. Now that we've talked about the various kinds of antimicrobials, now we need to discuss how we decide which ones to use. What we do is we test the various antimicrobials against a culture of the organism that's causing the disease. Or when we're first developing these antimicrobials, we'll test them against the toughest of the group or the most common of the group. Now, diffusion methods. You've already done one, the Kirby-Bauer test. This is where you put a lawn of bacteria onto agar, and then you take a paper disc that has been soaked with a known amount of the antimicrobial, drop it onto the agar, and the antimicrobial diffuses into the agar and inhibits the growth of the bacteria, or it doesn't. So looking here, you can tell that these two definitely don't inhibit the growth of the bacteria. But with these others, we would need to look at interpretation charts to be able to determine if the organism is resistant to that antimicrobial, and we shouldn't prescribe that antimicrobial for treatment of that organism, or if the organism is susceptible to that antimicrobial and you should prescribe it. Now, broth diffusion tests you haven't done. That's where we take 96 well plates and we put a known amount of microbe in, we put in various dilutions of the antimicrobial, and we see at what dilution the organism is prevented from growing. So if you look in here, you can see where there's some bacteria have grown and have sedimented into the bottom. Here we don't have any growth. So if this was, say, we'll pick this row. If this was going with most concentrated here, least concentrated here, right about here we would say was the minimum inhibitory concentration, or the MIC. So that is the concentration, that the minimum amount to inhibit the growth. You can see a little bit in there, but we'll pretend that there's not any. Here, the most dilute concentration is enough to inhibit the growth. Down here is even better. Nothing, nothing, nothing. Growth, growth. So right here would be your minimum inhibitory concentration. Here with disc diffusion tests, we can't tell the inhibitory concentration. We know the minimum inhibitory concentration is right here. Take a plug of the agar out to determine how much of the antibiotic is there at the border of the zone of inhibition. So what they've done is they've combined a diffusion method with a broth dilution method so that we can see both at the same time. What they do is they take these strips of paper and they soak various amounts of antimicrobials in there, and they put it down onto a lawn. And the higher the concentration, the further out it diffuses. Well, right here is the minimum inhibitory concentration, and we can read right off there how much antibiotic is on there. Now, we use this minimum inhibitory concentration to determine what dosage is going to be effective in controlling the bacteria. Later on, we'll talk about therapeutic index. This becomes important then. Now, speaking of resistance that we are determining from disc diffusion tests, or from minimum inhibitory concentrations, well, we're going to talk next about the mechanisms for how an organism develops resistance to antimicrobials, and we're going to talk about antibiotic misuse, which can lead to the development of resistance. Alright, there are four basic mechanisms by which a prokaryote can develop resistance to an antimicrobial. The first one here's our antimicrobial, is to not let it in the first place. You don't have transporter proteins to bring them in, or in the case of gram-negative bacteria, the antimicrobial has a tendency to get caught in between the outer membrane and the peptidoglycan cell wall. So it never gets in, never gets into its target. Now, another method that the organism can use to develop resistance is to change the shape of the target. So if this red doohickey here is, say, an enzyme that the antimicrobial is working against. If you develop a mutation where the antimicrobial doesn't fit as well and it doesn't competitively inhibit anymore, then the antimicrobial is going to be either not effective or less effective. So here's another one. If the organism has enzymes that break the antimicrobial, penicillinases are a prime example of this. Penicillinases go in, clip that beta-lactam ring, then your antimicrobial is no longer effective. Okay. Now the last one that we're going to talk about is some microbes, from our point of view, very annoyingly, have developed what are called super pumps. They are transporter proteins that as fast as antibiotics are brought into the cell, they're pumped right back out. It's kind of like a revolving door. And the reason why they're so annoying to us is because these super pumps, several of them pump out lots of antimicrobials. And that's where some of the microbes have developed multi-drug resistance. It's very annoying to us. It's a good strategy for the bacterium. Now realize with all of these resistant mechanisms have to come from a change in the DNA. It can either be a mutation in the chromosomal DNA, or they can acquire a plasmid that has a gene for, say, this super pump, or for a penicillinase, or for a different transporter protein that doesn't allow the antimicrobial to come in. So those are the two main ways of acquiring these changes. Now another thing to keep in mind before we leave resistance mechanisms is bacteria can't develop resistance because they want to. It's not like an antibiotic hits a population and some of them say, we've got to change, where's a plasmid? No, it's too late then. So you always have within a population of bacteria those organisms that already have resistance to various antimicrobials. They've had a random mutation and it hasn't hurt them. And then when the population is exposed to the antibiotic, then those organisms that are already resistant survive and increase in numbers because they don't have competition from the other members of their species that were in the population that were not resistant. Now, when you take antibiotics, the plan is for the antibiotics to take care of most of the microbes, and then your immune system takes care of the rest. Let's go on and talk about how antibiotic misuse can cause spread of resistant organisms within the human community. Here we see this lovely picture. In a lot of countries, you can go and get an antibiotic without a prescription. This is fairly common in lots of different countries. This has led to the spread of antibiotic resistance in a lot of bacterial populations because you're getting rid of susceptible organisms when you don't need to, when they're small in numbers and your immune system hasn't been stimulated to go and take them out. If you take an antibiotic for a viral infection, it's not going to do any good. You have to take an antiviral to be effective against viral. Antibiotics, usually, when we use that term, we mean things that are antibacterial. Lest we think that it's just people in other countries that are causing increases of the antibiotic-resistant microbes like methicillin-resistant Staphylococcus aureus or MRSA. Here in the United States, we are notorious for not taking the complete course of our antibiotics. We feel bad, we go to the doctor, they say, okay, you have strep throat, here's penicillin, and you take it and you start to feel better, and one, you think you don't need to take the drug anymore because you feel better, or two, you start forgetting because you feel better. Generally when we feel icky, then we're more prone to take the antibiotic. Well, if you don't take the complete course, the antibiotics don't kill as many of the microbes, and so your immune system has to take care of more of them. And your immune system is more likely to miss the resistant organisms. So take the complete course of your antibiotics. So I've already mentioned used for viral infections. They've done studies, and I forget the exact percentages, but it was something like half of all sore throats. The patients persist in insisting that they need a drug, and so the doctor just writes them out an antibiotic because it won't hurt. Unfortunately, this does lead to resistance within the various species of animal feeds. This one I'm kind of ambivalent on because I've never been a farmer, but I've lived around farmers, and their profit margin is very, very narrow. It doesn't take much to send them into bankruptcy. And if you put broad spectrum antibiotics into animal feeds, your animals put on weight and faster. And it's because they're not fighting off subclinical infections, and so they can put more of the energy from the food they're eating into building up muscle or laying more eggs. So, am I going to say, farmer, don't use antibiotics anymore? If it's a choice between bankruptcy or using antibiotics, if I were a farmer, I'd use antibiotics. Well, the problem is we've found an increase in resistance of microbes related to use of broad spectrum antibiotics in animal feeds. So now there's a trend toward selling antibiotic free animal products. They advertise them. They cost more. I'm willing to pay more, but not everybody can afford it. It's a problem, but it is one of those things that leads to increased resistance. Now let's talk about safety. As I mentioned before, we're going to talk about therapeutic index now. The therapeutic index is where you measure the toxic effect of the antimicrobial on the patient versus the therapeutic effect. In other words, the minimum inhibitory concentration. How much of the drug do you need to achieve the effect of killing or inhibiting the bacteria versus what nasty side effects you can get? In other words, is it going to start killing your mitochondria? Okay, so you have to balance the two. So we generally try to pick MICs that are well below the toxic effect of eukaryotic cells, and that would be the therapeutic index. Once you start bumping up against the toxic effect with the therapeutic effect, those are the antibiotics that usually we monitor very, very closely, and we would prefer to have you in the hospital so that we can monitor liver function, kidney function, see how well your mitochondria are holding up. Allergies. Probably you know somebody who's allergic to penicillin. You may be allergic yourself. There are also those who are allergic to sulfa drugs. You name the drug, there's somebody who's allergic to it. You have to keep this in mind when you prescribe these. Now, remember back to our chapter on hypersensitivities. Your immune system can take offense at some of these drugs and perceive them as being an indication of an infection. And so they cause an immune response even though the drug is trying to help you. So we generally don't give drugs to patients that are allergic to them. That's kind of a duh. Next, we're going to talk about synergy versus antagonism. Some drugs play well together and others do not. So let's look at some examples. Say you've got a drug like penicillin that inhibits cell wall synthesis. If you try to prescribe it with a drug that inhibits protein synthesis, say like tetracycline, they have a tendency to work against each other. The drug that inhibits protein synthesis slows down cell wall synthesis so you're not popping holes in the microbe. This is known as antagonism. They work against each other. If you prescribe those two drugs together, they will work worse than if you prescribed them separately. Now, say we take a cell wall synthesis drug and, like penicillin, and prescribe it, mix it with a penicillinase inhibitor. They help each other. They work better together than they did alone. We call this synergy. So that's basically synergy versus antagonism. This is also something you have to take into consideration when you're prescribing drugs. Which drugs have synergy and which have antagonism. Now for our last topic, the future of antimicrobials. We are constantly on the lookout for new antimicrobial agents, mostly because the microbes are constantly developing resistance to the antimicrobials that we have already. So we're developing new antibiotics. We're taking natural antibiotics that are produced by microbes. We're tweaking them a little bit to make semi-synthetic and synthetic antimicrobials. And all of this goes toward creating new antibiotics. Now, some of the things that we're excited about right now are antimicrobial peptides. These are very short chains of amino acids that are very similar to the antimicrobial peptides that you yourself produce. If we can produce them in large quantities in the lab and then prescribe them, that's giving your immune system a boost. That's exciting. Now on to antisense agents. Okay, I've got a very, very short mRNA, and in the normal course of things, the ribosome will clamp itself over the top of that, but ribosomes can only clamp over the top of single strands of RNA. Now let's say that for this same stretch of mRNA, which we'll say is a pathogen's mRNA, I produce a complementary strand of RNA, antisense RNA. So it binds to the single strand of mRNA. Now the ribosome can't get its mouth around it. Okay, the two strands of RNA together are too big to fit over the top. Now you can also do this with DNA because if you have two strands of DNA that will not come apart, then it can't be replicated or transcribed. So this is a way of targeting very specifically just the genetics of your pathogen, and it leaves the patient's DNA alone. This is also very exciting. Once again, we've come to the end of another exciting chapter. Make sure you go through this as many times as you would like. Come to class with questions. You know the drill.